Uh-huh. Very good. So, greetings and a couple of things. Uh, so, again, the exam is a week from today. Just a reminder, please, pencil. I only have a few spares. If those Scantrons, they don't, some of them do work with pen, ours do not. Then it becomes as a big zero and it screws up all the calculations I have to do for averages. So, make sure it's pencil. I have a pet peeve when someone marks with pen. If you don't have one, but I'll have a few extra. Hopefully, you will have one. So that and the other, like I said, maybe just a reminder. I realize the chem class that precedes us here frequently they don't evacuate real quick. I know it's okay. So I mean, if you need a few extra minutes, I don't want to go much more than be anything. But if the other students have a little more time than you do, I have to be fair to everybody. So I mean, I can always open up the lab then and kind of get you into it. I don't think Dr. Robbins would, because he's not in there until 3 o'clock anyway. So if you need a little because I'm still in this classroom the whole time. Nevertheless, I'm giving an exam to my other class in here anyway. So even then, because there aren't that many of them, you could probably just continue with the exam and they'll start theirs. Okay. Other note, I will not be here the day after the exam, the class day, the Friday after the exam, so that would be the third. So I have to be out of town for that period. I know you're disappointed, restraint. I don't want any tears, nor do I want any laughter. Uh, Nevertheless, so what will happen? Undoubtedly, I will have the grades done sometime over that weekend. They get posted on Blackboard. Here is the major pet peeve: do not ask me about points or percentages or anything like that. My only responsibility academically is to give out letter grades. That is all that I do. There will be a column, and I post them on. Will appear on your, however you access your grade register on Blackboard. It will show you. It will say, unless it's changed dramatically from this Blackboard Ultra, it'll say exam one with scale or whatever. Typically, I have to scale them or curve them to a certain extent. And in any event, that's what it will say, and there will be a letter grade. So if somebody tells you, oh, you have to have an 80%, I don't care. Okay, the letter grade that would be a minimum of 80% would be a B minus. So if you have to get a B in the class, B minus is a B as far as anybody else is concerned, vis-a-vis that these percentage things that people dwell on. And if you have to get a C in the class, then a C is a C or a C plus because there's no C minuses in this institution. Okay, you got it? Now, I mean, the letter grade might appear. I will use C minus as a grade, even D plus as a grade, for purposes of my grade calculations. But academically, below C, there's D, and that is sometimes advantageous for students who require a C because you can be pretty generous in that. Like if you got a skosh more than a D, you might bump it up to a C. Realistically. But again, understand whatever you need. That's important. So grades are going to become more of a concern. So I urge you to look at the reviews. I can tell how many people look at the reviews that I record on YouTube. Right now, it's like I realize you had a big thing over there to study for if you haven't taken it yet. There's a bunch there. I understand that. Avail yourself of this, please. Thank you very much. That's the informational part. On with the show. I want to finish up a little bit here and just sort of clarify because inevitably at the as I do often as we go through things, you tend to go rapidly as you get toward the end. So I want to basically address the whole idea of what is and why it's so important. The vast majority of things we move in and out of cells are surrounded in membrane. The name we give them is vesicles. We're either creating little pockets in the membrane and bringing it in, or we're taking those now what we've called vesicles, containing stuff, done something to it, and effectively spitting it out. Or sometimes we're making something inside the cell, containing it in membrane to protect it, and spitting it out. It's not as though your digestive tract cells, when you just had lunch, are going to shrink from all the membrane they're gobbling up. Membrane is conserved. So whatever you're bringing in, an equal amount of membrane is being spit out. So it maintains the shape of the cell. And part of the emphasis of today is to understand it's a very three-dimensional thing. Everything you see on a board is two-dimensional. The animations help, but the reality is that you have to look at it in that regard. So endo, bringing it in, whether it's solid, phagocytosis, liquid, oil, pinocytosis, they all involve receptors. What they address in this particular text as receptor-mediated endocytosis has relevance, and we'll get to it. And really, it has to do with almost like the genetic basis of certain kinds of receptors. And also, as you will find as we go on, unfortunately, a lot of bad guys, a lot of toxins, a lot of viruses, a lot of bacteria are able to, one of the ways they can be so nasty to us is they are able to basically hijack this mechanism and invade cells. And you get more of that in micro, but this is sort of creating the frame so you'll be able to understand it down the road. Because, other than folks who are in, let's say, biomedical engineering, by and large, if you're health sciences have to take micro, right? Yes? Yeah, okay. So you're stuck taking micro, which is my favorite course, interestingly, to teach. Though I probably have a vaster amount of knowledge for this one. And when we get rid of it, we spit it out. It's called exocytosis. It exits the cell. At least they're not classifying it based on what we're spitting out as much as we're taking it in. And the whole idea of transcytosis, and we see this in digestive and urinary, effectively the cell is just a boundary. And again, it's cased in membrane, so it goes through the cell because the inside of a cell can be a nasty place. And it basically traverses or goes through, transits through the cell, and then enters the bloodstream typically, or something called a lymph. And so in that case, it's really not impacting the cell. The cell is just, it's like somebody coming into class here and turning around, they got the wrong class now. They go. Pick an analogy that works for you. I'm having trouble creating one today. Let's see. So there are, not unlike receptors, all of these have proteins, usually receptors. It's very specific. Pathogens, bad guys. Patho is disease, gen is generator or causing. That's the derivation. That's there the same way when you look at pathophysiology, many of you in nursing, it'll be basically disease and understanding the physiology behind it. So some pathogens are capable of taking those receptors and making their way in. And anything that comes in, unless it's going through the cell via transcytosis, is always going to fuse with something called the lysosome. The lysosome has enzymes in it for digestion and defense. If it's something that's usable, the enzymes break it down into usable components, and it's either used inside the cell or processed and delivered to another location. It goes in the bloodstream to go to another cell that uses it more appropriately. If it's waste products, they get spit out by exocytosis. If it's bad guys, they get destroyed to their waste products, and it gets spit out, and eventually we excrete it. So that's what I mean by digestion and defense. We use enzymes are nasty. If you ever wondered why when you cut yourself, tore an ankle, anything, twisted an ankle, sprained something, stubbed a toe in the middle of the night, and you're like, oh my, like that. Cells have been disrupted. The lysosomes open up and they pour out these harsh enzyme chemicals. The enzymes are indiscriminate. They begin to destroy mostly proteins anywhere. It doesn't matter. They don't have to be inside the confines of the cell. That's why they hurt so much. And those chemicals play a very important role in cell signaling, which we're going to get to as well. A little bit today. This is all, it's all trying to present you with this sort of interesting role that cells and parts of cells have that are there. And here's just showing you, we looked at this before. It goes through. Sometimes we just get rid of it the way it is. Sometimes the lysosome interacts with it, all well and good. Don't have to go through these. Don't like these anyway that much. It's shrinking. And so phagocytosis, endocytosis, quote unquote, eating. And this is the whole idea of the sort of the fluidity and the three-dimensionality of the cell. You're going to see when we get into the parts of the cells, which is the next chapter, I just messaged you or emailed you with those PowerPoints from the remaining parts of this cell chapter, because they won't fit on the blackboard per se. So look for them in the mails as attachments. It's fairly obvious. where they are. Nevertheless, imagine you have things called microfilaments. They're basically made of tiny proteins that can assemble and disassemble. It's almost as though you had an arm here and it could magically disassemble and grow over here. And in doing so, it pushes on the membrane like this. And what happens is the membrane is pushed to one side, cytoplasm flows into it, and it's almost as though the cell moved. And so if you're pushing it on two parts of the membrane, the cytoplasm moves into them, they create those what we call pseudopods to engulf something. So it's all about things. We're constantly building things inside the cell and disassembling. Little organelles are moving inside the cell. It's not, get that notion out of your head that everything is in a fixed position. Some things are, but a lot of the other organelles have the ability to move, and other proteins will carry them to another location to do something else. And things are going on all the time. So I mean, the way I always frame it, when we see something two-dimensionally, it's static to us. But the reality, what being dynamic is, it's always changing. Just the nature of things, just the way you and I are changing constantly in different ways, whether we're aging, whether we're young, whether we're learning, whatever, observing. Pick an example, there's a zillion. So what happens is it forms this so-called phagosome where it engulfs it, whether it was solid or liquid, it's the same darn thing. And that's the amoeboid motion, these temporary extensions. They pull it in, and effectively, as the little proteins inside disappear, it looks like they're not there anymore. The reality is it's just redesigning the membrane can open and close. And they're showing you illustrations here. Usually the particles aren't this big. It doesn't necessarily attach to the receptors. It's sort of a change that allows these pseudopods, which means false feet, to form. And this is, now what's interesting about pinocytosis, and yeah, it really works the same way, membrane infolds, bring in the fluid, that's sampling of the environment. And really, in the small intestine, we've brought it down to fluid levels when it's brought in, and that's a very good way. And the idea of membrane components being recycled back, that's a good thing to recognize. It's the environmental sampling. And you're going to get an edge of my opinion as well. What our body does, what our immune system does, and you learned about it already in the skin, and maybe that's part of a test that you'll be taking from Dr. Robbins, or a quiz. Okay, you have all in the epidermis, you have keratinocytes, melanocytes, Merkel cells, and something called Langerhans cells or dendritic cells. Basically, dendritic means they have like lots and lots of tentacles, like trees when the leaves fall off. Those are little samplers. Anywhere we contact the outside environment, in your gut, in your skin, in any of these openings, right? We're constantly sampling the environment for our immune system to basically stay kind of tuned up. We're using it. Was that something I'm familiar with? Maybe we need to make an antibody to this. Maybe we have to construct a barrier. That's the beauty of that system. That's when they start talking about environmental sampling. It's really a way of using the immune system, and understand our bodies are all about use it or lose it. The absence of activity, the absence, whether it's mental, whether it's physical, any way you can construct it. And some people, I found that in my experience, having dealt with older people a lot over my professional career, not so much my teaching career, understand, the frame is my mom. Okay. She's beaten me in Scrabble when she's 86 years old. I was third. She was first. My sister, who was an English teacher by trade, before she got her MBA, second. I'm third. And I got a good vocabulary. My mom never went to college, but she read constantly. And she, probably until maybe 88 to 89 years old, she was totally kosher for Alzheimer's. Basically did her in, if you want to rephrase it that way. She lived to be 93. For the last 2 years, not a whole lot of recognition, unfortunately. But nevertheless, why am I mentioning this? There's something that you learn about, and I teach about it in micro. It's called the hygiene hypothesis. And it was a mistake that was made for the royalty didn't let their children play with other children. They scrubbed the walls. They didn't want any contact with the environment. It's crazy. They hadn't been exposed to regular diseases, things like that. And the classic example is the son of Henry VIII, who went through a number of wives to acquire said son by a lady named Jane Seymour, and he died before he was 16 years old from pneumonia. Okay, no contact with other kids. It's to some extent that's where my real feelings are. This is the opinion part. Vis-a-vis COVID, which I talk more about in micro. The whole idea of being constantly in masks, the whole idea of you folks not being able to interact. I mean, you weren't a terribly at-risk population. Let's be clear. Just weren't. It's okay. Overreaction. Again, Freeman's opinion, not fact. Nevertheless, this is the receptor-mediated endocytosis nugget. And yes, it can be transcytosis as well. They are highlighted by these things. You will not have to know this. If I ask you the question about clathrin-coated pits, believe me, the default answer is receptor-mediated endocytosis. This is a very specific way, and a lot of these, the clathrin, which is a protein, a very specific receptor, and particularly for these called low-density lipoproteins. Notice, insulin works this way, viruses, diphtheria, cholera. Diphtheria is a horrible disease. You are constantly being immunized for diphtheria. Why is that true? Because when you get a tetanus shot, you don't just get, it's not just to prevent tetanus. It's trivalent. Tetanus, diphtheria, pertussis, whooping cough. That's why we don't see a lot of diphtheria moving around. Okay. So all of those, the low density lipoproteins, are what people erroneously refer to as the bad cholesterol. You undoubtedly know somebody who takes what they call anti-cholesterol drugs or statins. Opinion coming. They are overprescribed, but the people who need them have a genetic defect that infects this system, and it doesn't allow these lipoproteins to be brought into the cell to be processed. When they're processed, they're made into useful things by the cell, as opposed to floating around in the bloodstream. If you don't have the receptors, they float in the bloodstream in large amounts. Whenever blood vessels are damaged, and if you did not know it, blood vessels are damaged all the time, areas of inflammation develop. It attracts those fatty molecules. The fatty molecules begin to calcify and harden. That's where we get something called athero or arteriosclerosis, hardening of the arteries. That's why people take those quote-unquote anti-cholesterol medications. Somebody you know is on statins. No question about it. That's why they're overprescribed, because they couldn't be that many cases. And we'll deal with caveolae and these small openings. We talk a little bit about smooth muscle. That's later in the show. That's not today. That's later in the semester. So I want you to take note of that. They show it to you here. And so basically it's creating, let's say, a very specific vesicle for a very specific purpose is the way I would describe it. And that's just a really good example of it. And that's the way I use it. Lastly, spitting it out. Thankfully, we don't have to classify it liquid, solids, or somewhere in between. It's all exocytosis, exiting the cell. See ya. Okay? And anything that we spit out and we make, it doesn't have to be stuff we brought in. It could be things that are manufactured by the cell. The classic example are things called neurotransmitters. Your nerves communicate with one another by chemicals. Your nerves tell your muscles to fire or not to fire by way of chemicals. Okay? They're called neurotransmitters. Because they are so easily broken down, they're encased in vesicles, membrane. If they're going to be spit out, we designate them as secretory. The secretion is what your cell produces, like mucus, like perspiration, like oils. Yes?
So whenever you were going over the it had like 8 things. You didn't know those.
Which ones are those?
It had it like after each slide. So it had like the endocytosis, the receptor mediated.
Yeah, what you should know is, just that endocytosis, bringing in phagocytosis, solids, pinocytosis, liquids or oils. And the receptor mediated is really about that answer with the LDLs, those lipoproteins. Remember that little nugget on each one.
I wasn't sure because you went over the slide, so I wasn't sure.
Yeah.
Okay.
So you're welcome. God bless you asked the question. I appreciate it. We're gonna have another opportunity to answer very shortly. Everybody, get ready to rumble. Okay. Exocytosis. And these are secretory vesicles. Okay. They're surrounded, and typically they come complete with this. This is a snare protein. It's snare because it engages another snare protein. This is one to help pull it. We call it a T snare. V for vesicle, T for transporting it. Okay. So what happens is they get close to each other, and there's a series of signals that activate this. They fuse together, hence the snare. They intertwine, and as they get close to, as it gets pulled to the membrane, the vesicle material in the membrane start to fuse, and it opens up a little opening called a fusion pore, and then voila, it spits it all out. Basically, it's not forcibly. It's just that the membrane now incorporates with the existing cell membrane and effectively expels it like so. Okay. This is extraordinarily important. Every time you spit out a neurotransmitter, it's going to make a nerve fire or a muscle fire. It's going to be a signal to something, or mucus to coat your throat or to coat your stomach. Pick a scenario. There's a zillion of them. Nevertheless, there's an agent. We take maybe the strongest biological toxin that we know of, refine it, take the tiniest amounts possible, and administer it to people selectively, because it's a neurotoxin that stops muscles from contracting. And we inject it into people therapeutically. And what it does is it blocks this part of the picture. It doesn't allow the snare proteins to fuse. And therefore it can't spit out whatever it's secreting. And specifically, it's something called acetylcholine at the neuromuscular junction. What chemical am I talking about? Yeah, somebody in each of the other two classes got it. I had to give them a hint. Are you ready for the hint? If I give you the hint and it's used cosmetically, would that jog your memory? Pardon me? Say it. Go ahead. Correct. This time you got it. 2 words, everybody, and not just you, because you got it right. Be courageous. There's no such thing as wrong. We learn as much from wrong answers as right answers. Please. That's my. If you ask, I don't tell tales out of school. If I'm telling my colleagues something, I'm going to tell you the same thing because it's about you. You guys don't ask many questions. That's why I make a big deal when someone asks me a question. You're not. And nobody in this classroom, including me, who knows everything. No way. As you get older, you learn how dumb you are, and in doing so, you get smarter. You learn what you don't know, and you understand what you do. Please. Nothing these last 74 years plus have taught me that. I was using the same line 10 years ago. I just changed the number. My wife said, "Are you still telling that same?" She uses a word that I don't want to repeat with TTY at the end of it. Story. Go ahead. No, I won't say it. Can't make me. Nevertheless, so that's Botox. It blocks that. We learn about that when we talk about the neuromuscular junction. So recall that. This is all the resting membrane potential stuff. We went over it. Look at the animation. Potassium is the player. Basically, the potassium plays a pivotal role. What's very interesting is it's all about those leak channels. We have more potassium ones. Effectively, what makes the inside of the cell more negative, okay, is that it's almost impossible. So the cell itself in the cytoplasm is neutral. But right along the membrane, that potassium can't neutralize all of the acidity from the proteins. Or proteins are made of amino acids, okay? And they're in water in the cell. So what happens is that C double bond OH. This is the chemistry. I just want to give you how it works. What an acid means is it donates a proton. So what you're left with after the proton separates is an H plus over here and a C double bond O with a minus sign. Okay? So proteins in water give up their protons, their hydrogen ions. Okay, and what's remained is a bunch of negatives. So, and the proteins are too big to leave the cell, so the interior of the cell right along that membrane is very negative. And even though there's a lot of potassium coming in, it can't overwhelm it. Okay, if you get the inside the middle of the cell, it's neutral. Everything is neutralized. If you're outside the cell, it's neutralized. But right along the membrane is where that membrane potential is. And what's important about that, cells that can have the capability of allowing that membrane potential to change rapidly are what we call reactive cells, nerve cells, and muscle cells. And the bulk of the physiology that we go over is in that. And inevitably, the students find that the most difficult thing. Okay? So here it is. Chloride is outside. That helps to offset the sodiums. Okay? We don't have chloride channels routinely. There are things that will cause chloride to enter the cell and make it more negative. That makes reactive cells less reactive. Famously, that's how alcohol works. Okay? It induces chloride. It has a mechanism of opening the cell and allowing more chloride in, or opening chloride, what we call chloride-gated channels that are there. But you can see inside this A with the minus sign, those are the proteins. The anion, unable to follow potassium through the membrane, they retain, and that's why we have the negativity just along the membrane itself. And that's the only part you really have to know from this. There's not a whole lot to say after it. Just the animation says it all. The big deal, since they're constantly moving, the potassium is always moving out and being reattractive to come back in. Okay, there's more potassium inside the cell, there's more sodium outside the cell. Even if the potassium moves out and the sodium moves in, it doesn't change because of those pumps. Even in a cell that's barely reactive, those sodium-potassium pumps are continuing to restore that gradient. That's why they're pumps and they require ATP. Effectively, they're pulling potassium back in to maintain that concentration gradient. They're pushing sodium back out to remain that concentration gradient, meaning you keep pulling potassium even though there's already potassium inside the cell to make sure it always comes out, if that makes any sense. But from a cell basis, that's how it works. The same thing with sodium coming in. And there's a little bit about these things. These have to do with interactions. The cell adhesion molecules are on the exam you're taking on Wednesday. It's basically definitional. You have desmosomes, which are the strongest. They have those plaques, they have those sort of zipper-like teeth, they have keratin structural filaments called intermediate filaments to reinforce it. They're all about holding the cells together. Gap junctions or connexons are proteins that have openings that allow either chemicals or electrical signals to pass. And the tight junctions, remember, all about watertight. They're basically holding the cells, cell to cells, closely together the more there are, so the stuff can't get in between the cells. We're not talking about getting through it, we're talking about getting in between it. Desmosomes, so they stay together firmly. Tight junctions, so stuff can't pass in between them. Connexons or gap junctions, so they can communicate. That's basically what you need to know for this. This is just giving you some idea of the role. And a lot of these are really, again, sugars and proteins. And we talked about some of this. It allows the cell to anchor to that matrix. It attaches those fibers you learned about in connective tissue. All the elastin, collagen, et cetera, et cetera. Cells, and this is dynamic, cells are always moving. That's how our immune system works inside. It's constantly rolling over things that are there. And this is what I mentioned before. When a cell is disrupted, it releases chemicals. Doesn't matter. It doesn't have to be cut. It could be a scrape, a bruise, a twist. Inevitably, microscopically, cells are disrupted. That's why it hurts. It hurts because the cell releases chemicals contained in lysosomes. Some of those chemicals act as what we call, it's a fancy name, cytokines. Talk about that extensively in AP2 and immune system. Kines means moving, cyto means cell. And basically, white blood cells are always around and on the move. In a lot of places, they have large, we call pools of them, areas where they're stored more than others. And these chemical signals And it's not like they know where they're going. It's a chemical signal. They're either attracted or repelled. In this case, they're attracted. They are attracted and move. The chemical is attached to something. Of course, the cell will move without going crazy about it. So those cytokines are what bring white blood cells to where you cut yourself and why it gets inflamed and why it stops bleeding and why there are healing factors and why it swells up. All that's it's by design. Again, when it goes out of control, that's a problem. So, and some things are worse than others. I got a nice chunk out of my leg from not watching where I was going over the summer. It's been 2 months. It's still sitting here. Thankfully, it healed, it's effectively healing, albeit slowly, without incident. So they attract white blood cells. Cytokinesis is the fancy term. The animation I have will show you that when we get to it, not today. And again, all of these different junctions, you can make more, you can break them down, and signals directing which way cells are going to go. We don't understand what makes one cell proliferate, why something will change into a particular kind of a tissue. That's sort of where the forefront of research. We're trying to grow organs and stuff like that. Things like cord blood, having the ability. Did you talk about that at all in mesenchymal cells and when you talk about connective tissue? Are you familiar with the idea of collecting cord blood? When a baby is born, the umbilical cord has we have a fancy name, pluripotential. Pluri means it could become literally anything. Okay, and that's a quote unquote, that's a true stem cell. You have stem cell lines. So theoretically, people will save this hoping that, and typically will freeze it and bank it for the person's lifetime or their child's lifetime. If down the road we figure out how to make a new liver or make a new kidney, that type of stuff. What they used to call organ banks and things like that, as opposed to relying on transplants. So if something has not developed properly, you might choose to inject cord blood in an effort to improve these things. This is often done with children who have had problems early in life. So you were not familiar with cord blood infusions or that type of stuff? Okay. Interesting thing to read. More on that down the road. See, you learn something new every day. Contact signaling. And then really, again, the surface is what happens as the cells roll over and contact each other. It helps them develop and helps them become what they're supposed to be. And yeah, lots of chemicals opening all these different channels. That's when we'll learn about these transmitters and hormones. And in the immune system, we talk about paracrines. Para means next to or alongside of. So these are chemicals that work it's like working on the same cell or the cell next to it. We'll get to that in the immune system when you do AP2. And not a lot to say. This is a little confusing. Okay, I mentioned before that it's kind of how I looked at signal chain. Insulin kind of works this way. Human growth hormone works this way. Again, it's a giant protein. You can't get it in the cell. It attaches to something called, we call it the primary messenger. It enters the cell. We see the term ligand on the illustration on the left here. The ligand is too large. It doesn't look that way from here, but imagine it's very, very long. Contact the receptor. We call that the first messenger. Effectively, it sets up a chain reaction because it changes the shape of the receptor. It sets up a chain reaction on the interior, resulting in something called a second messenger to generate this sort of cascade effect that's there. You're going to see this a number of times. You don't have to go crazy about this now. Why they call it a G protein? Remember, our basic energy is ATP. GTP, which is part of this system, G stands for guanine, just like the A's and T's, the G's and the C's. It is very similar to adenine because they're both purines. So it shouldn't surprise you that where we have ATP and have GTP, they're very, very similar in their chemical construction. So this is another one of those. GTP is 3-phosphates. It's got a couple of high energy phosphate bonds, just like ATP. Interestingly enough, what happens during this pathway is it gets the energy from this GTP that you see on the illustration. And interestingly enough, ATP becomes involved because ATP loses two of its phosphates, those two high energy phosphates, and is left with something called AMP. And that's one phosphate. That's just a pure nucleotide, adenosine monophosphate. And what happens, it has this sort of interesting ability to kind of make a little circle out of itself. So one of the big second messengers that does all the stuff, this cascade of events inside, so it's called cyclic AMP. Another one, your old friend calcium, ionic calcium. You'll see this a lot down the road. This is, again, contextual because you probably see this, and it's like they're totally like a relay race. Here from the outside causes a change on the inside. That causes a change in this protein, and then a change in that protein, and etc. All the way down to some final protein, which is some kind of an enzyme, and that triggers that last reaction, activates the second messenger, again, either cyclic AMP or calcium, typically in these cells. And then you have this sort of cascading event. And when you see a kinase means it's taken this high energy phosphates and caused that to activate a system. So even though the insulin is opening the door for glucose, it's also generating a force for the glucose to be used to create energy. It's advising the cell to increase mitosis. It's telling the cell not to utilize the HGH, human growth hormone pathways, etc., etc. So there's a lot that goes on inside. And again, you're not going to have to remember all of these names. Just to understand the complexity of it. And it's the way. Basically, there's two ways that things can impact what goes on inside the cell. If they're water soluble and they're basically large like this, they work through this mechanism. If they're fat soluble and small enough, not giant, they can go right through the membrane and actually contact receptors inside the cell and directly impact genetic mechanisms, transcription and translation. You're going to see this when you start to get to the endocrine system in AP2. So you're getting a heads up on it now. And then there's nothing left, thankfully, in this section. Good. We get to introduce the next one. Guess where we're at? That's right. Glad you mentioned parts of the cell. Oh, we had this already. I learned parts of the cell ages ago. Not my way. Okay. The R part. Yes, the ribosome makes proteins. Yes, the nucleus has DNA and RNA and blah, 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 genetic. All that's true. But understand how the interlinkage of all of these. And yeah, probably the way that I would, not this exam, but the subsequent exams, they have, just the answer key you can see here, would probably be these 5 things, and I have a little statement, you just match them up. Not hard. But I need to explain in a little bit more detail how it works. First, cytoplasm or cytosol. What makes it a plasm, it's got a combination of liquid, a solution, basically we call it sol gel, has areas where it's solution, liquid, gel, where there's more protein, like jello or gelatin. And the more gelatin, the firmer it gets. Or it has things that are inherently solid, inclusions, molecules that are in there that don't dissolve, and glycogen, energy storage, pigments. Very often neurotransmitters have pigmentation. Fat droplets, again, storage for potential energy, things like that. And obviously the organelles, which are almost like a mini cell unto itself, particularly if they're the ones that are membrane bound. So they're very, very specialized, and that's what we're going to look at. So understand, they're not all membrane bound organelles. For instance, the ribosome, by definition, is not membrane bound. It receives a signal when it starts to make a protein. The messenger RNA, which we're going to look at, is the thing that carries that instruction to the ribosome. there's a molecule, there's like a part of that messenger RNA that basically says, oh, we can't make that here. We've got to make it because it's going to be bound in membrane. It's going to be one of those vesicles we talked about. And you'll see where they go as we get through this particular presentation, not today, but later. And basically, it's going to say, okay, you're going to go over and bind yourself to one of these membranes to something called the endoplasmic reticulum, because we've got to make this protein a particular way. Sometimes it's just going to make it right at the ribosome in the middle of the cytoplasm. That's called a free ribosome. So ribosomes, by definition, are not membrane bound, but sometimes they become attached to the membrane if they're doing something relatively specialized. That's the kind of detail that I have to start to acquaint you with. Are you ever going to use this? Most people in here, no. Why do we have to know about it? I don't know. It's part of the curriculum. But it really gives you an idea of the role of the cell. Mitochondria, everybody knows that's the energy factory. And we'll get to this. All this talk about membranes, they're part of something called the endomembrane system. It starts at the nuclear membrane and protects things in membrane, so that the inside of the cell won't alter or destroy them. And we'll get to these things. The Golgi apparatus is the packing and shipping department inside the cell. And here, if I want to send something out, I send a book out to one of the high schools that's working with us. I send it to the mailroom. Give an order, send it here, blah, 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 blah. Boop. I don't have to, it wouldn't be hard for me to do it, but they take care of it, and our department pays for it. Just like you might send a package out, except you might take it to the post office after you pack it and tape everything together. Peroxisomes are like lysosomes, except they deal with toxic derivatives of oxygen. You didn't know oxygen could be a bad guy. And we'll go into all of these things. So this is where structure and function are married. This is where you get all that. I'm going all crazy about how the fan pulls things. First of all, the glucose that comes into your cell before it gets started broken down, for it to maximize its ATP production, it's got to have oxygen. The good news, trouble is, too much oxygen is bad. We have mechanisms to deal with excessive amounts of oxygen. We did not know it. That's where the peroxisomes are going to come into play. Okay. Here, form and function. You have an outer membrane that's permeable, and the inner membrane is a series of folds to create surface area. We'll see that in a second. So we're able to create a tremendous amount of ATP generation in a tiny space. And moreover, when we need to make more of these, we can, because they have their own DNA and their own RNA. You're going to learn in micro, those are actually identical is maybe the wrong term, highly similar to bacterial DNA and RNA, bacterial DNA and ribosomes. Something you learn about called endosymbiotic theory. That's what makes our cells, we have components that mimic. We replicate by mitosis or meiosis. Bacteria just keep dividing in two. It's called fission. So that's all that's coming for you in micro. And so this is what it looks like. All this double membrane, all this is surface area. It's embedded with proteins acting as enzymes. And because of that, it's able to take this pre-processed sugar and in the aid of oxygen, effectively create large amounts of energy. Okay? The oxygen is sort of the ticket to get it in. And again, sometimes excesses of oxygen are problematic. So it's the key, and I'll leave you with this reaction you're going to see a lot. We call this cellular respiration. What respiration is basically energy and giving off waste product. So the energy comes from, we'll use glucose for an example, that's sort of the universal C6H12O6, plus oxygen to balance the equation. And what it comes to, and I'm putting it reversible for the reason, releases carbon dioxide and oxygen and water, H2O as waste products. We easily get rid of it. We exhale carbon dioxide, we urinate, we respire, we get rid of water, a lot of it. And Because of that elegant structure with all those folds, able to generate large amount of ATP. That's what respiration is. When it's a cellular basis, it's exactly what it is. Pulmonary involves more of the exchange of oxygen and carbon dioxide from the waste product out and good oxygen coming in. The reverse of this happens all the time too. It's the basis where we acquire most of our sugars. We take energy, not ATP, but solar energy, in the presence of water, the atmosphere, carbon dioxide in the atmosphere, with the help of a useful enzyme that are in plants called chlorophyll. And what do we make? Carbohydrates. As a bonus, oxygen. Photosynthesis. And so it's interesting how the oxygen we get to breathe on this planet comes from chlorophyll. The vast majority of it, by the way, comes not from what you're seeing out here, though that from what we're breathing, but from the ocean, something called algae. But the planet's 5/6 covered in water. That really limited amount of this good, nice looking green stuff we have here, at least until the winter is upon us. That is photosynthesis. So when you go home or outside, thank a plant, thank a tree. Thank you very much for providing me with oxygen and carbohydrates I need for energy transformation. So I'll leave you with that. Thank you very much. Catch everybody.